So, Valerie and back. And I would remind everyone, since Valerie announced Philip's book, that it is coming out in December, just in time for Christmas, correct?
You bet, and you get a box of chocolates for that one. Yes, it will be out in time for Christmas.
So you know what my wife is getting for Christmas. So now your question.
This question's for Dr. Rusche. In the latest shareholder report that I received from Repligen, they indicated that they are looking for another pharmaceutical company to take over the HDAC inhibitor research. So my question is, have you found someone? And if you don't, are you going to keep this going full steam ahead?
We are looking for additional resources to put on these programs. They're very expensive and for a small company, very difficult to maintain. Also, Repligen is focusing a lot of its efforts in a different area than this disease at the present time. So it's imperative that we do find additional resources. We have not slacked on what we're doing and what we're trying to do, and hope that over the next year we'll be successful at bringing additional resources to the program.
So you don't have anybody yet, you're still looking?
I'm sorry.
The question is, do you have anybody yet?
No, we don't. Simple answer.
Cassie in the middle.
Don't kill me, Dr. Lynch, but I have a question about the variability of the echocardiograms, because we started the echocardiograms with you guys at CHOP when Albert was in phase 3 idebenone. And then, of course, we live in New York, so we have a cardiologist at home, and he's had echocardiograms, done in New York since. So isn't there something called a Z-score that will kind of take, the variability of the different echoes and kind of make it more concrete?
The question is about variability in echocardiograms and whether they can be adapted for a Z-score. Yes, that can be adapted for what's called a Z-score, so you compare it to age-dependent controls. The age-dependent norms are different in different locations. It's also, though, somewhat technique-dependent. And for example, I'll just give an example. If you cross from one side of the street, CHOP, to the other side of the street, Hospital of the University of Pennsylvania, pediatrics, adults. They, in fact, use the functional outcome of how much systolic dysfunction a person has. They use different numbers. Fractional shortening is commonly used in kids. Ejection fraction is used in adults. And they both have their merits as well as their disadvantages. There are fundamental aspects to that which simply differ. There are technique issues. It is, for example, in young children, it's hard to get a really long echocardiogram so that you can get the complex calculations. That's what Mark St. John Sutton explained to me once. So that's part of it, is that variability. The other aspect is that these numbers, we don't know what they mean in terms of long-term prognostication. Most likely the hypertrophy, and this has been proven in cellular models as well as in people, is a reactive process. It happens because you have mitochondrial dysfunction. So if it gets better, does that mean that you made the mitochondrial dysfunction worse? Or did it all of a sudden fail and things are getting better because they were pseudo-normalized and they're in fact getting worse and your heart just isn't as thick for that reason? This is the problematic aspect of echo which none of us have figured out how to deal with. Kim made reference to strain imaging this morning, which is basically taking a simple echo and doing much more calculated calculations on it. Off the record, it probably works in FA because it gets rid of the biphasic nature. That said, there are no norms for kids. So all of them also are both regular echoes and strain imaging are heavily normalized to body surface area. And very frequently, we all cheat on whether we use body surface area versus body mass index and things like that. That's part of the variability. Yes?
You were alluding to the fact that you typically have to be careful with us as a group because we try to, as best we can, hold you to what you say. You had said, hang on, and in October, there would be some things coming first, second quarter of 2013 that you were super excited about. So I guess I'm, towards the end of the day wondering what's coming. What do we anticipate? What'd you find out?
So the question essentially is what clinical trials are on the horizon, and particularly very near horizon at this moment. Remember, I am not allowed to comment on any clinical trial or its beginning until it's officially approved by our IRB, unless it is commented on by someone else outside our organization. If you were watching the University of South Florida Symposium, Dr. Miller did mention that there will be a follow-on study of EPI 743, which is a close sister compound is a good phrase. Almost identical twins, but just subtle differences. Dr. Miller, one patient told me today that he had been told by Dr. Miller that December would be the time when he might expect it to begin. Dr. Miller did mention publicly that the primary site was the University of South Florida, and there would be sites at the Children's Hospital of Philadelphia and the University of California, Los Angeles. Remember that the A0001 study, as we published in July, is a very exciting result. The issue is that 1 month, our bodies are all very smart. Whether you have Friedreich ataxia or not, we will adapt to whatever we get. For example, I take so much caffeine, it does not do anything to me anymore. If you get a little bit of A0001 or EPI 743 for a month, it could well make you better, but how long does that benefit last? The answer is, I don't know. Let's just do the experiment and find out. Yes.
Okay. My question is for Jim Rusche. With the follow-on compound for, I think it was 3250, do you know if the FDA would be open to using the data from the Italian trial to maybe go ahead and start in patients for that one? And then the other part is, do you have an overall timeline for the follow-up compound?
That's a very good question, and one that we're going to put to them this fall. I think it's highly likely that the FDA will take into account the fact that there is an HDAC inhibitor that has been in Friedreich's patients before. It's relevant information. And as I said, the real challenge is to be able to discern safety in a beginning trial like that. And what we would like to suggest is that the study in Italy demonstrates that that's possible. We'll have that discussion later this fall with the FDA, and that will clarify exactly what we do to prepare for a clinical study sometime in the next year.
I would mention, get the back to Cassie, but for EPI 743 versus EPI 0001, there are 6 hydrocarbons different. You can use it as supportive data, but it cannot replace certain studies for the most part. 6 hydrocarbons is still a regulatory difference. They're not perfectly interchangeable. Cassie.
I was just wondering, since we're talking about study and participation, are you making any headway in changing some of the possible exclusionary criteria for FAers to be included in the study? For example, being able to walk 25 feet, which was the one in idebenone, and for things that are coming down the pike. I mean, obviously, if you had diabetes, and that could possibly be, a different distinction. So I know that there's all kinds of variables, but as far as like the walking, seems like it would exclude so many FAers who would be willing to participate. It just seems like it just needs to be taken care of, Dave Lynch.
Very good question, Cassie. The question is, in essence, how are inclusion and exclusion criteria decided upon? Remembering that any clinical trial is an experiment. The goal is to test your therapeutic compound or gene therapy or whatever, and give it the best possible chance for success for the least possible chance for variability, while realizing that there are only 5,000 people with FA in the United States. It thus depends very much on sample size calculations and the relative responsiveness of individuals at different points in the disorder. In the idebenone phase 3, you had to be able to walk. That was an inclusion criteria which was decided upon by Santhera based on their phase 2 data, when they flogged it a little extra to see if they had a certain responsiveness. That is in contradiction to the rate of change of people on the FARS scale and who are walking and not walking. They change at the same rate. Now, the question of biological responsiveness is one which is unique for any compound or therapy. There will be some things which would be highly likely to work at any stage in the disease, and others which would be much more likely to work early, and conceivably other things which would be more likely to work late. Then you have the measurability aspect. For example, if I were doing a study on vision, I would target the people with more significant disease because they've lost more letters on that torture we call the low contrast letter acuity chart, and thus we'll be able to measure more improvement if they got better. That's common sense. They sent me to school to learn that. These are the things that play in. Let me assure you that if anyone writes a trial or study which we do not think will recruit successfully the required number of individuals, we will have an extended discussion with them as to why and what they might do to expand their inclusion criteria so that they get enough people without changing accessibility. And that's ASSESSIBIL to you. Yes, Norm. Haven't had a question from you in a while.
Ever since the triathlon, he's been slowing down. James, the question is to you. In your dealings with the FDA, when I'm in pharmaceutical development at a baseline bench stuff through Columbia University, so I've had some experience. My question, what you said today is like big pharma trying to bring a drug in that's going to potentially affect millions of people. The drugs that we're working on is actually very limited, should be orphan drugs, and I think we've got the status according to everybody I've talked to. Do you find them to be open, that we've got a limited patient population, and that the regulations can be, are we thinking out of the box with them? I'm just trying to get a feedback for the rest of the audience too of what's going on at that level, because this is where our frustration is coming from. We want something tomorrow. We are, as families, willing to take a little bit more of a risk. The FDA, of course, is there to protect us. So you see my dilemma.
Yeah.
We warned you they would be very sophisticated, Jim.
Yeah, no, it's I think, very much to the point. And in our development, it's easy to get frustrated that things don't move very quickly, but there is a need to be careful in movement forward and to be able to assess accurately the safety and pharmacology of a drug. If you don't, you end up wasting a lot of time and potentially people's lives. So that's a real important role. What I think we don't do well enough with the agency, and there needs to be a general recognition of that by everybody, the agency, companies, and the advocacy groups. We don't work together pragmatically to find solutions to the ends of the spectrum. All safety means it takes you forever to accomplish a goal and check every box that doesn't take into account pragmatically, this is a orphan population with no current treatments and a patient population that's sophisticated enough to participate in trials and help find safety, pharmacology, and efficacy. So, as we were discussing earlier today, that's an area where I don't think that we all work together and do a good enough job to think outside the box, as you say, but to, for example, come up with novel clinical trial designs that can accomplish more with less patients, less time, less money, which means more testing would get done. Companies like mine would find it easier to participate in this space if they could afford to. So we have to do better at that.
Bridget, and then Preston. Oh, Preston first. No. Yeah.
Yeah, this is a kind of a follow-up, I think, to Norm's question. I'm looking at this from an engineer's perspective of how do we apply this, how do we make sense of it, kind of a common sense thing. Frustration that I have that maybe some other people share is with a progressive debilitating disorder, why do we have to deal with placebos? Because even a lot of the data that you showed up there, Dave, it seems to me there should be a statistical way to take the normal variation and normalize it somehow. So, what's the point of placebos in a progressive disorder that we know statistically gets worse?
The question is, do we need placebos? I'll give a perfect example. The Idebenone 1999 result is now largely questioned based on two reasons. The selection of that population for a very highly hypertrophic, as well as the lack of placebo to directly compare it to. Placebo treatments, there are situations where they are not necessary. When your treatment is invasive and can't really be mimicked. You could do it all with natural history data. However, you're going to have it so statistically different that you slam dunk it. The variability of Friedreich ataxia is so significant that we would have to increase our sample sizes hugely. Or the trial which Edison has announced they were planning on EPI 743. Notice I've gotten the whole announcement in without saying that we did it. Is it planned to include 60 individuals using no placebo, and of which one group will be placebo. That's public information. If we didn't use that to have a firm comparison group, knowing the variability of FA and just taking our natural history data, which are not equivalent to placebo dosing, because in every neurologic disease, we see a placebo effect, and if you don't, you should worry about your study. If we didn't have a placebo group, you'd have to triple or quadruple the size of that because of the huge variability in FA. Now that gets to the question, in essence, that Jim was being asked about funds. Human experimentation is expensive. Going back to Giovanni's model systems, worms are not expensive. Human experimentation is quite expensive, even when it is a natural history study. Believe me, you don't want to know the budget that our clinical network runs every year. But it is worth every dollar because it allows us to limit the size of the therapeutic intervention, the people needed for therapeutic intervention. Yes?
Pam.
Oh.
Pam, in the back.
Back. Oh, Pam.
Yeah, as Dave's pointed out, looking forward is, sure. She wants to know how long it will take to finish the study in Italy of the HDAC inhibitor. So yes, we're through the first two cohorts. The next two cohorts were originally designed with a study design of crossover, which would take much longer. But we're looking at a study operation and some changes to that protocol that would allow us to complete it over the next 6 months. And we're hoping to be able to do that. We have a similar group of very dedicated people in northern Italy that have helped that study progress. Very soon after we have them. A week. All of the data will be compiled. We have to assure that there is no mistakes in the data, so there's quality control. That usually takes another month. So I would expect sometime shortly after the end of the first quarter of next year.
Dr. Lynch.
Oh, okay, Bridget.
So here's my question. Are we gonna talk a little bit about idebenone and how it is off like the FDA and that are arguing?
Bridget has asked about the current regulatory status of idebenone. And that is a perfect question for us to handle in the question and answer period is the first thing after we finish after Giovanni's last talk. So we will handle that then because there are some developments and some things we will be able to present to you. I want to thank everyone. I know Jim has to get on the road. Thank you very much for coming down.
I have one question. How fast would Dr. Lynch have talked this afternoon had he not ridden 50 miles yesterday?
Faster.
Fast enough to power a small New England city is the old phrase. Thank you all, and we'll take a break and then be right back with Travel Path.